let us now start with the first segment, which is the Young Scientist presentations. We invite Prof. Liu Xiaogang from the Singapore University of Technology and Design to chair the session, and our presenter, Awais Kasim. Regrettably, another of our young scientists, Dr. Rachael Lappan, is unable to join us in person today, but she has kindly prepared a video recording. So I'll now hand over the session to Prof. Liu, please.
Okay, ladies and gentlemen, a very good morning. My name is Xiaogang. I'm from Singapore University Technology and Design. I'm working on chemistry research, specifically computational chemistry. And today it's my honor to be the chair for this morning session. So welcome to Singapore, welcome to SUTD. I seldom have a conference starting at 8 a.m. It's really early, so you are really the early birds and you are awesome. Okay, so our first speaker will be Dr. Rachael Lappan. Rachael is now working as a research fellow at the Monash University in Australia. Her research is about microbiology. Specifically, she's studying how microbial is living in the harsh environment and how they interact with our world. Microbial are very small, but the impact could be big. So let's watch the video from Dr. Lappan.
Okay, hello everyone. My name is Rachael Lappan. I'm a postdoc at Monash University, which is in Melbourne in Australia. And even though I can't be here in person today, I'm recording this from my hotel room. I'm really excited to share with you this new research that I will be embarking on this year about microbial life in the atmosphere. But I do want to begin by talking about microbes in general and also how some of my previous work led me to this really exciting area. So we really are living on a microbial planet. Microbes are essentially the foundation of all ecosystems on Earth because in our guts and in the guts of other animals, they aid in digestion and metabolism, they assist the functioning of our immune system, and they help us to defend ourselves against pathogens. In the world's oceans, microbes are making up most of the biomass there, and they play important roles in decomposition and photosynthesis, so really forming an important part of the carbon cycle. And they have similarly important roles in soil, particularly where they form close relationships with plants. Microbes are also inherently connected to health. Not only human health, but also animal and environmental health. So these three areas shown down on the bottom left are really linked and dependent on each other, which is a concept referred to as One Health, the idea that you need to treat issues in these areas in a holistic way. And some of the earlier work that I did as a postdoc focused on pathogen surveillance in this project called RISE, which is still ongoing. And so here we were aiming to improve water and sanitation in informal settlements with some new infrastructure. And I was involved in developing techniques to keep track of the many different diarrheal pathogens across humans and animals and various aspects of the environment. So doing this work really sort of, I guess, opened my eyes and taught me about the connections between human and animal and environmental health, this One Health idea. But something that I find really exciting about microbiology is how microbes can live in these very unique places where nothing else can. They can live at extremely hot temperatures, in hot springs, they can be in very acidic conditions, and also in the cold and dry valleys of Antarctica. There are microbes living where you would think that life couldn't be. And so this really, this interest really brought me to the field of microbial ecology, which looks at the microbial communities that live in a certain place, how they interact with each other, and also how they interact and are really shaped by their environment. So when it comes to studying microbial ecology, we need to do things fairly differently to traditional microbiology, because a lot of microbiology is based on culture-dependent work, which means that we have to grow the microbes in the lab before we can study them. However, the majority of the microbes out in the environment, in most places, in fact, and especially in these more unusual places on the planet, are uncultivated, which means that we just haven't yet worked out how to get them to grow in the lab, and they may not like to grow that way. So especially when we're looking at an entire community of microbes, and there might be hundreds of species there, this is where metagenomics comes in. So we've talked about DNA sequencing so far in this conference. So instead of growing the microbes with metagenomics, we're looking at their DNA. And metagenomics takes all of the DNA from a sample like soil or water, and we sequence everything so that you can look at the genetic information of every organism that is in there, all of the microbes and anything else. And so this tells us two very important things. It tells us firstly about taxonomy, or the profile of who is there in the community and how abundant they are compared to other species. And it also tells us about their function, because in doing all of this DNA sequencing, we can look at the genes that they have, and that tells us about the sorts of things that they are capable of doing. And so what I want to say here is that my real sort of passion now is doing work to understand how microbes can survive in challenging environments. The idea that, you know, life is sort of living in places where it really shouldn't be, but it finds a way nonetheless. And the reason why I find this so exciting is because microbes are extremely metabolically flexible. And what I mean by this is that we generally eat carbohydrates and we respire oxygen. We sort of don't have any other options. We can't really live any other way, and we would die without either of those things. But so we're not very flexible. But microbes can eat all sorts of organic and inorganic compounds to get their energy, including atmospheric gases. And they can also respire oxygen like we do, or they can live without it, live anaerobically and respire something else like nitrate or sulfate instead. And so different organisms can use different strategies for survival, but it's also quite common for any given organism to be able to use multiple survival strategies depending on what is available in their environment. So they're very flexible, they're adaptable. And so I'm really fascinated by this, and particularly, as I mentioned, the ability for microbes to use atmospheric trace gases as their source of energy. And this is the focus of the lab group that I work in, and there's been some really exciting advances here in recent years. So I'm going to talk about that a little bit. So as you will know, the atmosphere is mostly nitrogen and oxygen, which does a great job at supporting aerobic life. But there's also a lot of other gases in the atmosphere that are present in trace amounts. And these gases, particularly hydrogen, carbon monoxide, and methane, have recently emerged as this sort of hidden energy source for microbes. It's only recently that it's become appreciated just how widespread and important these gases, widespread the ability to use them, and how important the gases are, these trace amounts in the atmosphere, in sustaining a lot of different microbial communities. And much of the work that our lab has done has focused on how microbes can use these gases, particularly those three that I mentioned, as their source of energy. And so one of the places where this is really important is in Antarctica. So photosynthesis, which would usually be the main call it primary production, you know, where most of the biomass comes from, it's really challenging to do photosynthesis in Antarctica because it's a process that requires water, and Antarctica is actually very dry for parts of the year. There is also no sunlight, and the soils in these Antarctic deserts are actually quite nutrient limited in general. And so we've found that there's actually this huge diversity of microbes, and in fact the majority of the community, you can see the trace gas oxidizer abundance down there, is actually capable of using the small amounts of hydrogen and carbon monoxide from the atmosphere to sustain themselves. So many of the members of these microbial communities are essentially living on thin air. And so in the greening lab, where I work as a postdoc, I have worked across a number of different projects looking at how microbes can live on air in different places. So one of these was in desert soils. So I looked in hot and cold deserts, and we found that hydrogen is a really important energy source in the desert. We've done these experiments that you can see where we sort of very sensitively measure the amount of atmospheric hydrogen that is consumed over time by the microbes in these soils. I've also worked on a project in termite mounds where termites actually, they produce a lot of methane and also hydrogen. And we found that the microbes within the termite mound were actually consuming some of these gases released by the termites. And so to some extent, the microbes are mitigating the emissions of methane and certainly of hydrogen. There's a net hydrogen uptake by the mound. And that's how the termites are sort of feeding the microbes in these mounds and this interesting relationship between them. And I've also worked in the marine space where we found that even in the ocean, dissolved carbon monoxide and hydrogen are actually a really important energy source for microbes in the ocean as well. And the microbes that have the ability to use these gases for energy become more abundant in the ocean the deeper you go. You can see that in the graph for both carbon monoxide and hydrogen. And so in each of these projects, it was really the metagenomic approach that allowed us to explore these microbial survival strategies. And so that brings me to how these sort of seemingly very different projects have kind of come together. And, you know, it's come from me doing this work and being around other research going on in the lab about extreme environments and thinking about this, you know, continually wild idea that bacteria can eat gases to get their energy. And then also with me coming from this sort of One Health and disease surveillance kind of background, I was thinking about all of that, and I came across the field of atmospheric microbiology, as in microbes living up in the atmosphere. And I thought, wow, this is really something that's very exciting. It's an area that has not been very well explored at all, at least not in a metagenomics sense, because most of the research about bioaerosols in the atmosphere is about pollen or allergens. There's a lot of papers that are focused on how microbes are dispersed from one place to another by the atmosphere. But there's not that many research groups in the world that are focusing on microbial communities that are actually living in the atmosphere. And so this led me to be thinking, is the atmosphere actually just another extreme environment on the planet that actually hosts this substantial and diverse ecosystem, perhaps, of microbes? And so I took this idea and I applied for and received this early career fellowship that we have in Australia, which really allows me to sort of independently embark on this journey of exploring this research area more, where this combination of my experience and expertise that I built over the past few years is what's really sort of led to my perspectives here. So thinking about the atmosphere as a microbial ecosystem, like I said, there's not a great deal of literature here. The atmosphere has certainly been investigated for microbes, and there is some good work out there, but much of it has really sort of considered microbes to just be transiently moved from one place to another by the atmosphere, and that's it. So this idea that there could be microbes that are calling the atmosphere home, you know, being sort of resilient to the harsh conditions up there, and even perhaps actively doing something in the atmosphere, is still quite a very new idea. And I think one of the reasons why this has not been explored very much, or at least not nearly as much as these entire fields of soil microbiology or marine microbiology, is because it's a really challenging thing to study. And so why is that? So firstly, if you want to look at things with metagenomics, it's not something that existed until about 20 years ago. So this sort of world of opportunities that have only recently opened up in order to study this. The other major issue, as you can imagine, is that there's a very low amount of biomass in air. You think about taking an air sample, if you grab a bag of air, there's really essentially nothing in there. So if you're attempting to sort of very sensitively detect microbes and look at their genomes and, look at the entire community that could be up there, it's very hard to do this without contamination from other sources becoming a significant issue. So I have started some work here. Like I said at the beginning, this is a new area of research for me across the next 3 years. This is really what I'm going to be focusing on. But I have made a start, and I'm essentially just taking all of the precautions that I can when I'm sampling. That's a photo of me. So I dress as though I'm working in a clean room when I'm sampling to minimize the amount of cells and dust and stuff that's coming off of me. And then I will be extracting the DNA in this clean room facility that we have at Monash University. Not this one's at NASA, although it might be a bit cheeky for me to say I'm pretty sure the one that we've got is actually cleaner. So really just taking all of the precautions that I can to prevent contamination from getting into the samples in the first place is really the key, and doing that at every opportunity. So where can I actually go with this research? So obviously the very first thing to do is to really overcome those challenges of maximizing the amount of biomass that we can sample, minimizing contamination. And then given that that's sort of what's almost held up the field this whole time, is once that's overcome, there are so many different ways that all of this can go. So these 3 aims are what I've laid out for my fellowship. I really want to get a thorough understanding of exactly what microbes are up in this microbial ecosystem. And indeed, if it is an ecosystem, I want to understand, secondly, whether they are metabolically active and what they're doing. Are they performing important functions like perhaps regulating the composition of the atmosphere or degrading certain compounds? And then I also want to investigate the role of atmospheric microbes in seeding new environments on land, like in glacier forelands. How do these brand new ecosystems start in an area that was previously barren? So that's what I'm planning to do. But this kind of work really has a lot of connections that I can see, and probably many that I haven't thought of. So firstly, it takes me back to my One Health experience, because the atmosphere is critical in the processes of airborne pathogen transmission, antimicrobial resistance, dissemination of that over potentially large scales, which obviously has health significance. It's really important in understanding, better understanding global biodiversity, how the atmospheric microbes interact with those on land and how things change depending on where you are in the world. Regarding the microbial activities in the atmosphere, if you think about it, the atmosphere is covering literally the entire surface of the planet. So it's mass, or maybe not mass, but volume-wise, it's the largest potential microbial ecosystem on the planet. So if there are microbes there that are carrying out important functions like they do in our oceans and in our soils, their impact is probably actually quite critical to the planet. So I really want to look into that. And finally, of course, this work also contributes to our understanding of the limits of life. Just how far can microbes go to survive, both in the context of our planet and potentially on others as well. So I will leave it here by simply summarizing that microbes have and always will shape our planet. They are the dominant life form. We probably wouldn't survive long without them, and they'll be here long after we're gone, I expect. My experience in a couple of seemingly different areas is what led me down this exciting avenue of research, where there is really a lot yet to learn. And once we've overcome those technical challenges, then there are so many avenues for some very exciting research questions yet to come. And I just want to say that interdisciplinary science really is the way forward. Us talking to each other from extremely different fields is how the innovation and everything comes about. So I do welcome your ideas and your questions. I want to acknowledge the funding sources for this work that I'm embarking on, Chris Greening and the Greening Lab. And I'm very sorry that I couldn't be there in person. So please reach out to me on Twitter or by email I do want to hear your thoughts, your insights, your questions. I am absolutely happy to answer anything. Thank you so much for your attention today.